I want to introduce our next speaker, the last speaker of the session this morning, Dr. Alvaro Garcia Guerra from the Ohio State University, so I don't get in trouble with people that must have that. And he'll be presenting a talk today on the endocrine control of follicle selection in ruminants, a tale of multiple ovulations in cattle.
Thanks, Laura. I learned it the hard way that I needed to say the Ohio State University. So, all right. Well, good morning, everyone, and thank you for the opportunity to present data to you here this morning. And what I'm going to talk to you about is the mechanism of ovarian follicle selection and some data particularly relating to an abnormal phenotype, which is the selection of multiple follicles and consequently multiple ovulations in cattle. The majority of the data I'm going to show to you today is actually started during my doctoral program at UW-Madison, working under Dr. Milo Wilbank, and it actually continues as a collaborative project today, particularly under the multi-state research group, the NE 1727. So, as most of you know, the ovarian follicle selection process is a mechanism by which a single follicle from a cohort of growing follicles is selected and is allowed to continue to grow and ultimately acquire the capacity to ovulate. This process has been studied to great length in single or normal wild type single ovulating cattle. And we know that 2 to 3 days after follicle wave emergence, that single follicle is selected and is allowed to continue to grow while the other follicles in the what we call the follicular wave begin to regress and eventually undergo atresia and disappear. The morphological visualization of the selection mechanism has been actually called diameter deviation, and is basically defined as a relatively abrupt change in growth rate between the future dominant follicle here on the top and the subordinate follicle. And so basically what happens is the future dominant follicle is allowed to continue to grow at a relatively similar rate, while the subordinate follicles begin to first slow that growth rate and eventually completely cease to grow. We know that that diameter deviation occurs between 2 to 3 days after follicular wave emergence. And in Bos terrace cattle, the future dominant follicles is 8.5 millimeters in diameter. And that's fairly consistent. The other thing that we've noticed, or that has been extensively researched, is that that diameter deviation coincides with FSH reaching its native concentration, which, as I will talk about more later on, it's a critical component of preventing the selection of additional follicles. So as you can see, a great deal has been learned about this process by studying just normal single ovulating cattle. We also know that the time of diameter deviation, there's a lot of changes within that future dominant follicle that we call the dominant follicle phenotype, by which there's changes in granulosa cells, such as acquisition of LH receptors, increased expression of PAPPA, aromatase, and the consequent increase in estradiol. And in fact, it's this increase in estradiol that has been shown to be responsible for providing final suppression of FSH so it can reach its needed concentrations and prevent the selection of additional dominant follicles. As with any biological process, it's relatively interesting or a useful tool to use abnormal phenotypes to study these mechanisms. And those, in the case of follicle selection in a monovular species, would be the occurrence of multiple ovulations. So there are several models in ruminants, starting with the high fecundity alleles in sheep, the USDA marked twinner cattle, or the high producing lactating dairy cow. And in fact, the high fecundity genotypes in sheep have been a very interesting model because they have highlighted or discovered new mechanisms that were previously unknown that worked on the regulation of granulosa cell function. And then those was actually the TGF-beta family members that were discovered that had a key function in regulating the proliferation of granulosa cells. So there's about 19 different mutations in sheep that lead to multiple ovulations and multiple births. And last time I checked, there's always some new ones coming up, but about 80% of them are mutations are present on members of the TGF-beta family, basically BMP15 and GDF9 of one of the receptors, the BMPR1B, which was actually the original mutation discovered, commonly known as the borula. What these mutations cause is basically an increase in ovulation rate in the heterozygous state, and it can lead to even further increases in ovulation rate when present on the homozygous state. However, in certain lines, when these mutations are present in the homozygous state, they lead to primary ovarian failure, which is basically the cessation of folliculogenesis at the primary or secondary stage. So we have learned quite a bit from these mutations in sheep. One of the issues that we have is that it's very difficult to do dynamic studies on the ovary by ultrasonography in sheep for a variety of reasons that I'm not going to get into right now. But based on these, the knowledge of the role of TGF-beta on these genotypes, several models have been proposed in which oocyte-secreted factors, which are basically BMP15 and GDF9, will act upon the receptors on granulosa cells, stimulate activation of SMAD pathways that ultimately lead to increased proliferation of granulosa cells and inhibition of their differentiation. So what happens in these high genotypes is the factors themselves, BMP15 and GDF9, are mutated. Basically, a lot of these are loss of function mutations, or the receptor itself is mutated, which leads to reduced signaling of these SMAD pathways and the consequent reduction in granulosa cell proliferation and an increase in their differentiation. So until very recently, there had been no high fecundity genotypes discovered in cattle. And it was in 1996 that a highly fecund cow named Treble, that is shown there in the picture, was actually discovered in New Zealand, and she had 3 consecutive sets of triplets in her life. Although those cows would usually be culled, this farmer was really interested in it and actually kept it because in most of those calving events, all 3 triplets were actually born live and were able to survive. So what they did was grab one of the sons of this cow named Trio, which is on the black baldy calf here in the background, and then here is on a mature bull, and use that bull to actually inseminate females, and their daughters show multiple births as well, indicating the transmission of a genetic factor across generations. Dr. Brian Kirkpatrick, professor at UW-Madison, had been searching for a mutation like this for his entire life. And so he actually imported semen of Trio into the United States and used it at the UW-Madison herd. And Trio was found to produce daughters that had multiple ovulations in multiple births. So then they went on to do some of the genetic analysis, and this led to evidence for segregation of a major gene affecting ovulation rate that was located in chromosome 10, and more importantly, it did not coincide with any of the previously identified mutations in sheep, meaning that we were actually looking at a new mutation that would contribute to ovulation rate. So before we go into some of the detailed studies, I wanted to show you what we actually mean by multiple ovulations. So here we actually have data from cattle that we examine ovulation rate by counting corporeal lutein multiple cycles. On the top part, you can see the frequency distribution. So non-carrier animals are our control single ovulating animals. You can see that in 90% of their cycles, they have single ovulations. And like we always know, there's some animals that will have eventually double ovulations, but no triple or quadruple ovulations. In the case of our carrier animals, in this case, we're heterozygous for the mutation. We have that 70% of the cycles have 3 or 4 ovulations, with only about 5% of those cycles having single ovulations. And in average, we can see 3.5 ovulations per cycle versus 1.1 in our non-carrier controls. Through a lot of work in doing a lot of superovulation and embryo transfer, we were able to generate the first few homozygous animals for the mutation. And what we saw evaluated ovulation rate by counting corporeal lutein, you can see that on average, they have about 4 ovulations, which interestingly, and if you remember what I mentioned previously about the sheep mutations, through this data, we could conclude that the homozygous state of the Trio allele did not lead to primary ovarian failure, which was one of the hallmarks of some of the other mutations. So over the last several years, we've been trying to answer the question is, what are the mechanisms that explain the high ovulation rate? In other words, how is the follicle selection mechanism altered in order to allow the selection of multiple dominant follicles that will eventually lead to multiple ovulations and multiple births? And there's 2 things I want to point out before we go into the details, is that most of the studies I'm going to show you right now use heterozygous Trio carriers as our group of interest, and then also that our control group are actually age-matched half siblings of the trio carriers, meaning we were trying to control the genetic background of our control group as well. So the first thing we asked was a very crude and simple question. There's been this theory that greater number of follicles will somehow contribute to the increase in ovulation rate. Although this seems overly simplistic because we still have a selection mechanism, the idea was that a greater number of follicles would actually basically override the selection mechanism and allow for multiple follicles to become dominant and eventually ovulate. So in order to evaluate this, we took a very simple approach, which was actually to do what we call antral follicle count, which is nothing more than counting antral follicles via ultrasonography at the onset of the wave. And we also did it indirectly by measuring anti-Mullerian hormone, which, as you know, in females is produced by the granulosa cells and is highly correlated with antral follicle count. So these were done in two separate studies. Antral follicle count was done at the onset of the wave, and AMH was done as a mean AMH of 2 samples during a synchronized follicular wave. As you can see, despite variation in antral follicle count and AMH, we saw no difference between the two genotypes. Taking this one step further, we wanted to see that even within a genotype, if there was any relationship between the antral follicle count and ovulation rate. And we had concurrent data on the follicle counts and the ovulation rate for multiple cycles in some of these animals. So you can see here in the scatter plot that neither in the non-carriers or the trio carrier animals, despite significant variation in antral follicle count, there was no association with ovulation rate. Even in those trio carriers that had relatively high ovulation rates of 8 ovulations per cycle, there was no association with the follicle count. So our conclusion was that antral follicle count or the ovarian follicle population had really no role in the selection of multidominant follicles, at least in these genotype. So then we went on to look at what was the actual molecular change. If you remember, the original genetic studies had indicated that the causative polymorphism for the trio allele was in chromosome 10, and it spanned a region of about 1.2 megabases, and there was 3 positional candidates in that region. SMAD6, SMAD3, both of them which are part of the TGF-beta signaling pathway, and then IQCH, which had been associated with age at menarche in women. So we did a study where we actually collected through ovarectomy dominant follicles that were previously synchronized, and we took the granulosa cells and subject them to RNA sequencing. Here, I'm only going to show you the data on the 3 positional candidates, but what we found was that SMAD6 was actually overexpressed in the granulosa cells of trio carriers compared to their single ovulating control animals. We later then went to confirm this by collecting granulosa cells of follicles of different sizes and measured SMAD6 expression via qRT-PCR. And we found that, first of all, there was no relationship or association with follicle size, meaning that SMAD6 expression did not change with follicle size, and that overall there was about a 9-fold greater expression of SMAD6 in trio carriers compared to the non-carrier animals. So it appeared that SMAD6 was actually the key molecular change. So how does this fit with the cellular model that we had before? So as I said, BMP15 and GDF9 act on those granulosa cells and stimulate the SMAD pathways. And actually, SMAD6 is a negative modulator, or the SMAD158 pathway intracellular. So the idea would be that trio carriers, by having an overexpression of SMAD6, they will have dampening of the signal of that SMAD158 pathway, leading to reduced granulosa cell proliferation. So a very similar mechanism of what we have seen in the mutations in sheep. So we ask ourselves, based on this, 2 questions: is follicle growth, reduced follicle growth involved in selection of multiple follicles? And then, as I'll explain a little bit later on, we also wanted to look at FSH because of the role of FSH in providing ability to select multiple dominant follicles. So the first thing we did was actually looked at follicle dynamics, and this is a lot of what we do, is basically try to evaluate follicle growth from a dynamics perspective using ultrasonography. And we usually take a rather complicated approach, but basically we synchronize follicular waves and we try to control the endocrine environment, particularly progesterone, as much as we can. And then what we did was evaluate by ultrasonography and take collected serum samples for FSH every 12 hours until those animals ovulated. So here's the growth pattern of follicles for trio carriers and non-carriers. In the case of trio carriers, I'm going to show you the most common patterns always, because remember, they don't have always the same number of ovulations, so it gets kind of complicated. But there's two things I want to highlight from this image. First of all, you probably noticed that follicles in trio carriers are smaller than in non-carrier cattle. And then the second one is, despite the fact that they have multiple dominant follicles, there is still a selection mechanism as shown by this subordinate follicle here. So meaning that not all the follicles in the wave actually get selected and become dominant. So the selection mechanism is still present. And if that was the case, then we could calculate diameter deviation, which remember was that change in growth rate that you see around here. So we calculated diameter deviation and we found that it occurs at the same time after wave emergence, right around day 3 in both genotypes. And then we also were able to establish the mean follicle diameter at the time of deviation. And we see that trio carrier follicles have much smaller size follicles at the time of deviation. We also looked at pre-ovulatory follicles, and we see that trio carriers have about 4 ovulations per cycle. And then when we look at follicle size, we see that no matter which one we take in the trio carriers, which is whether it's the smallest one, the largest one, or the average, they're always smaller than in the single ovulating animals. But perhaps the more interesting thing was when we looked at ovulatory follicle volume, assuming the follicle is a sphere, we see a very dramatic difference. But the more interesting part is that you see that trio carriers have 4 ovulations, right around 4 ovulations per cycle, but the ovulatory follicles are about 1/4 in size compared to what we see in single ovulating animals. So then that brought us to the question whether follicle diameter was really the right way to go and assess the functionality of follicles. And it's a very intuitive way to do it in diameter because we use ultrasonography and you're looking at a 2-dimensional screen. However, as obvious as it may seem, the follicle is a 3-dimensional structure. So we decided to start thinking about follicle functionality from that perspective. So if we go back to our data, the pre-ovulatory follicle in non-carrier single ovulating animals was about 1,300 cubic millimeters. When we look at our trio carriers, they had about 4 follicles, each one of them of about 310 cubic millimeters. If you add the volume of those together, assuming that they would still function as a single unit, you see that the total follicle volume of those pre-ovulatory follicles was about the same. So when we go back and look at the follicle growth profiles from a volume perspective, always assuming that the follicle is a sphere. So you see on the right-hand side, mean follicle diameter for both genotypes, you see they're different. And when we express it as mean dominant follicle volume, you also see that follicles in trio carriers are smaller. However, when we combine the dominant follicles and add the volume of each individual follicle, you can see that total dominant follicle volume was not different between the 2 genotypes. So with this in mind, and knowing that actually, despite the follicles being smaller, the time from follicular wave emergence until they actually ovulate was not different, then we decided to look at growth rate. So here you have growth rate based on diameter. So in millimeters per day, you see that trio carriers grow follicles more slowly. And when you express it on a volume basis, we keep coming up with this relationship in which trio carrier follicles actually grow at about 1/4 of the rate of what a single follicle would in our non-carrier control animals. So the idea being here that follicles in trio carriers develop at about a 1/4 of the rate. They end up being about 1/4 of the size of single ovulating animals. That's we need 4 follicles in order to have the same total volume at the end of the process. So with this in mind, we thought, well, now the next logical step was actually to look at the resulting corpora luteum. Remember, the follicles ovulate and produce a corpus luteum. So we wanted to see whether each individual CL was smaller. And however, when we added all the luteal tissue together, where it would actually still produce the same total luteal tissue as if you were to have a single corpora lutea. So here you have individual CL volume for both genotypes. And you can see trio carriers have much smaller CL. And then, however, when we combine all the individual CLs on these multiple ovulation animals, they equate to the same total luteal tissue volume. And as a result, their hormonal output measured as progesterone is the same between the 2 genotypes. So again, follicles and CL in trio carriers are individually smaller, but multiple are required to obtain the same total luteal volume and the same hormonal or the same total volume, whether you're talking about follicles or CL, and the same hormonal output as in single ovulating animals. The next thing we wanted to look at was FSH. And this comes because FSH is heavily involved in the selection mechanism. Remember, it has to reach needed concentrations right when the selection or diameter deviation is actually occurring. And then also because we know that providing exogenous FSH, we can induce rescue of the subordinate follicles and induce multiple ovulations, which is a common strategy used in assisted reproductive technologies for embryo transfer. So it was logical to think that FSH would actually be playing a role. So we, throughout the same experiment I just showed you, we measure circulating FSH, and overall FSH was greater on average for the entire period. However, when we looked at specific time point comparisons, we saw tendencies for greater FSH right around the time when deviation, when that selection actually is occurring. So then we decided to look at average FSH in very defined periods. We looked at pre-deviation, at deviation, and post-deviation. We saw that a pre-deviation, which would be the top part of the top of the FSH surge, concentrations were not different between the two genotypes. And after deviation, so the nadir was actually not different either, but the time period of 24 hours encompassing the time when deviation occurs, FSH was actually greater in the trio carriers, which led us to believe that a delay in the decrease in circulating FSH would allow trio carriers to actually select multiple follicles. If we go back to the kind of the original slide that I had at the very beginning, you'll remember that I said that at the time of deviation, there's a lot of intrafollicular changes in that future dominant follicle, both in the granulosa cells and the resulting concentration of hormones inside the follicular fluid. So based on that, we wanted to ask ourselves two questions. Would follicle dominance, or that dominant phenotype that we see, is acquired at a smaller size? That would be reasonable to expect based on what I just showed you just on the growth profiles. And also we wanted to see is once that phenotype is acquired, would actually be similar in both genotypes. So in order to do that, we actually took two different approaches. The first one was to evaluate ovulatory response after providing a large ovulatory stimulus through injection of GnRH. The reason we do this is because the culprit or the hallmark of dominance is the acquisition of ovulatory capacity. The second approach we did was actually collecting granulosa cells and follicular fluid from follicles and actually evaluating some of those markers that had been previously identified for dominance, whether it was through gene expression for granulosa cells and follicular fluid hormones. So here's the first study in which we evaluated ovulatory capacity. So we synchronized the emergence of a follicular wave in both our genotypes, and then we challenge animals at different days after follicle wave emergence with 200 micrograms of gonadal acetate, which is a fairly large stimulus for causing ovulation. What we see is not a large sample size, but we see that in timing, there was really no differences in the proportion of animals that ovulated at each of the different days after wave emergence. But remember that the main question we wanted to ask ourselves was, what was the minimum follicle size at which ovulatory capacity was attained? So in order to do that, we group all the different follicles of the different days and evaluated the probability of ovulation as a function of follicle size and genotype. As you would expect, as follicle size increases, the probability of ovulation increases as well. However, you can see that the curve in the trio carriers is shifted to the left, indicating that ovulatory capacity is acquired at a smaller size. When we take the 50% probability point, we can see that on a diameter basis, follicles in trio carriers are about 5.5 millimeters, where non-carrier single ovulating animals are about 8.3, which is usually the time where diameter deviation has been detected. And when we put it in a volume basis, we see that single ovulating animals acquire ovulatory capacity when they're about 3.5 fold greater than in trio carriers. So we keep coming with this 3 to 4 fold relationship as we have seen before. So the conclusion was that trio carriers acquire ovulatory capacity at a similar time after wave emergence. However, they do so when they're about 1/3 to 1/4 of the size as in a single non-carrier ovulating animals. So then we went on to look at the intrafollicular markers. So very similar approach. We synchronize the emergence of a follicular wave, and then we come in and aspirate each individual follicle. So each one was 3 to 4 follicles, depending on whether they were single ovulating animals or multiple ovulating animals. And we collect each follicle individual, both follicular fluid and the granulosa cells by transvaginal ultrasound-guided aspiration. And when we run a series of hormones, I'm not going to show you everything that we've done in some markers or mRNA expression of some of the most common markers of dominance in the granulosa cells by QRT-PCR. So here's some of the data. As you can see on the left panel, estradiol concentration is probably the most consistent marker that has been found in multiple studies for dominance. And we know that estradiol concentrations will increase as follicle size increases. But as you can probably appreciate here, the first significant increase occurred at a smaller size in trio carriers between 5 and 6 millimeters, while in the non-carriers that happened between 7 and 8. And then a very similar picture was found in the expression of LH receptor in granulosa cells. Again, we see an effective follicle size in both the genotypes, but in the trio carriers, that first significant increase occurred at a much smaller size. So then we took a slightly different approach, and we were able to, through the use of estradiol concentrations, we were able to develop a cutoff that allowed us to classify follicles into whether they were dominant or non-dominant. And that when it took us back to the same approach that I showed you a few minutes ago with the ovulatory capacity, in which we were able to evaluate the probability of dominance as a function of follicle size and genotype. And we did that both for estradiol, for LH receptor, and for several other factors. And we always come up with the same type of graph, basically a relationship. Follicle size has a relationship with the probability of dominance. So bigger they are, the more likely they're going to be dominant. But always that curve is shifted to the left in the case of trio carriers, indicating they acquire dominance at a smaller size. If we take again the 50% probability point, you see that on a diameter basis, follicles in trio carriers are smaller, as I said. And when you put it on a volume basis, we always come with this relationship in which single ovulating animals acquire dominance when follicles are about 3 to 3.5 fold greater in size. So just like we saw with the ovulatory capacity, trio carriers will acquire dominance at about 1/3 to 1/4 of the size of non-carriers. So then we wanted to see, okay, well, if they do acquire dominance at a smaller size, but what about the actual phenotype of those dominant follicles? So we classified follicles in whether dominant or non-dominant, irrespective of size, and we actually looked at several of these markers. Here you have estradiol. You can see a significant increase from predominant to dominant, which is something we knew now for many, many years, but again, no difference between the two genotypes once you account for their stage. And then same with aromatase, as you would expect, there's an increase in the expression of aromatase, which leads eventually to the increase in estradiol. And again, no difference between the two genotypes at either the predominant or the dominant state. We also looked at PAPPA, which, if you remember from one of the talks yesterday, is an IGF binding protein protease that has been associated with the acquisition of dominance. And we see again that it increases from the predominant to the dominant stage. However, no difference between the two genotypes. And then a similar picture for LH receptor, which is perhaps one of the most critical markers for dominance. The only did find that in the predominant stage, there's slightly greater, significant, but slightly greater expression of LH receptor in trio carrier follicles. So with this, we could conclude that although the dominant phenotype is acquired at a smaller size in trio carriers, the actual phenotype once dominance acquired is not really different between the two genotypes. The other really interesting approach that has been utilized is the ablation of LH support using GnRH antagonists. So here you can see in the top panel here on the left, the LH pulses in a control animal, about 9 pulses in a 10-hour period. If you treat them with acetylene, which is a very power GnRH antagonist, you ablate those pulses completely, and then we are able to replace LH action by using human chorionic gonadotropin, although we do it in a non-pulsatile manner because HCG has a very long half-life. This approach has been used extensively in single ovulating animals, and what happens is if you take away LH support, diameter deviation doesn't occur. There's no selection of a dominant follicle, and the maximum follicle size attained is actually the size at which dominance would normally be acquired in those animals, if that kind of makes sense. So we actually did this study where we took both genotypes and randomized them through 4 different treatments: a control and treating control group, a group with the GnRH antagonist acetylene, a group with human chorionic gonadotropin only, and then a group with acylin plus the HCG. So because there's a lot of groups and panels here, I'm going to show you the single ovulating or non-carrier animals first. You see here the control group. They have diameter deviation and selection of a single dominant follicle, as you would expect, really not the same picture that we always see. When we treat those animals with acylin, we prevent diameter deviation and selection of a dominant follicle, and the maximum follicle size attained is right around 8.5 millimeters, which is when we would normally expect those follicles to become dominant. When we restore LH support through the addition of HCG in the acylin-treated animals, we see that we restore diameter deviation and selection of a single dominant follicle, all about occurring at the same time points. And then the HCG group alone really did not differ from the control. They still have diameter deviation at the same time and selection of a single dominant follicle. So what happens in the trio carriers? So here I'm going to remember that we have different patterns. We have animals that select 2, 3, or 4 dominant follicles. I'm going to show you the most common one. So keep in mind those sample sizes at the top will show you the proportion of the animals that we're depicting. So our control animal, in this case, the majority of the heifers selected 2 dominant follicles. You can see that diameter deviation occurs. However, that diameter deviation occurs at a smaller follicle size as we had seen before. When we gave acylin, just like in the controls, we prevent diameter deviation, we prevent selection of a dominant follicle, and the maximal follicle size attained is 6.4 millimeters, which, if you remember, one of the first study was when we had actually calculated the diameter deviation occurred in trio carriers, so much smaller than in the single ovulating animals. When you look at the acylin plus HCG, just like in the control group, we were able to restore diameter deviation and selection of dominant follicle and at a similar time. However, the surprising part was 6 out of 8 of the animals actually selected only 1 dominant follicle, which was very interesting. And then our HCG group really did not differ very much from the control. They still had diameter deviation. They selected either 2 or 3 dominant follicles, very much like in the control. The only difference was that their final size for those dominant follicles is slightly larger than in the control group. So this brought us to the idea that, as we knew for single ovulating animals, LH is absolutely required for acquisition of dominance, despite being acquired at a smaller size. And perhaps the more interesting part was that the role of GnRH-induced LH, or even perhaps GnRH-induced FSH pulses, was required for the selection of multiple dominant follicles. The other thing we wanted to actually look is whether the dominant phenotype in those follicles in trio carriers was actually acquired in a sequential manner. And what we mean by that is that as the wave develops, the first follicle in the hierarchy would acquire the dominant phenotype first, followed by the second one and the third one and the fourth one, and so on and so forth. So what we actually did was, going back to one of the studies where we collected follicular fluid, we were able to classify heifers, determine the percentage of heifers at different days that had either 0, 1, 2, or 3 or more dominant follicles. So if you look here on the left panel, you see that on day 2, which is the first row here, the majority of the animals have 0 dominant follicles, and a few animals have already started going through selection, so they have a single dominant follicle. As time goes by, you see that the percentage of animals that have no dominant follicle goes down and disappears, as you would expect. And then the majority of animals will end up having a single dominant follicle, although we do have a few that will select 2. However, when we look at the trio carriers, the interesting part was that on day 2, you see that the majority of animals have no dominant follicles, just like in the non-carrier group. However, of the ones that do have a dominant follicle, is only a single one instead of multiple ones. And as we start going through days, you see that just like in the controls, the percentage of animals with no dominant follicle decreases. And then we have in the initial days a distribution with animals that have a single, 2, or 3 dominant follicles. But by the time we get to day 4 and 5, the majority of animals have multiple dominant follicles, which led us to the conclusion that our hypothesis was correct in the sense that trio follicles acquire dominance in a sequential manner rather than all of them at exactly the same time. So the last piece of information I wanted to show you that is still kind of a work in progress is we wanted to look at the contribution of each of those smaller follicles to the regulation of FSH. If you remember from the very beginning, I mentioned that FSH reaching those native concentrations was critical to prevent those subordinate follicles from continuing to grow and acquire dominance. So the idea was that because the true carrier animals have smaller follicles, although their intrafollicular estradiol is the same concentration, the hormonal output would be of each single follicle would be less. And so if we took some of those follicles away, we would change the regulation of FSH. So what we ended up doing is using what we call a one follicle model. We synchronize follicular wave, and 5 days later, when we know that dominance has already been acquired, we randomize animals of both treatment groups to either one follicle model or our sham control. And basically the one follicle model, as you see in the picture below, what it means is physically by ultrasound guided aspiration, removing every single follicle that is 3 millimeters or more, except one of the presumptive dominant follicles. So we do it based on size. In the single ovulating animals was relatively easy because the size difference by that time of the wave is really huge. And in the trio carriers, what we did was took the largest one, assuming that that one was going to be the dominant, one of the dominant follicles, which is usually, of course, the case. And then we actually monitor follicle growth and circulating FSH. Because in the sake of time, I'm only going to show you the FSH concentrations. So there on the left, you see a picture of what the ovary looks like after we do that. So you only see a couple very, very small follicles in this ovary, and then just a single dominant follicle here. And we actually normalize FSH concentrations to pre-treatment levels, so they're expressed as a percentage change. And what you can see is that as soon as we create that one follicle model in the trio carrier, FSH rises and becomes significantly elevated compared to the other groups. And we really did not see much change in trio carrier controls or in the non-carriers that had a single dominant follicle. It didn't really matter whether we took the other ones away. So that gave us the premise that FSH, after deviation occurs, once you have selection of dominant follicles, FSH is inhibited by the single dominant follicle in the case of control animals, while multiple dominant follicles are required in the case of the trio carriers. So where we are, and this I promise I'll finish here shortly, what we have is basically based on a lot of the data we have from the sheep models, we know that the two oocyte secreted factors are important components in the regulation of granulosa cell function, and they both stimulate granulosa cell proliferation. From a whole animal perspective, we know that a follicular wave begins in response to the surge in FSH. During that initial part of the wave, all follicles grow at about the same rate, and they all contribute through inhibiting to the decline of FSH. And 2 to 3 days after that, a single dominant follicle is usually selected. That implies all those changes in the follicle, acquisition of LH receptors, and so on and so forth, that lead to the increase in the production of estradiol, which actually provides the final suppression of FSH, preventing other follicles from acquiring that dominant follicle phenotype. And through some of the studies using acylene and HCG to replace, we know that LH support is absolutely essential for that selection mechanism, although we were able to show that whether that LH support is in the form of pulses or in a constant pattern, you still have selection of a single dominant follicle. So in our trio carriers, what is happening is we know through some of the initial studies that the key molecular change appears to be the increase in SMAD6 expression, which remember is a negative modulator of the SMAD signaling pathway. So we hypothesize it will lead to reduced granulosa cell proliferation, although this still needs to be tested in a cell culture system. And from a whole animal perspective, we know that a follicular wave in trio carriers start the same way after a surge in FSH. However, those follicles already from the onset of the wave, they're about half the size of those in non-carrier animals. They develop at a slower growth rate. So by the time we get to day 2 or 3, they're about 25 to 30% of the size of those in control animals. And at that point, the first follicle on the hierarchy will acquire that dominant phenotype, acquisition of LH receptors, increasing estradiol production. However, because it's smaller in size, the amount of estradiol output into the circulation is lower. And thus, it does not completely suppress FSH. And it allows the next follicle in the hierarchy to acquire a dominant phenotype, contributing itself to the production of estradiol and keep decreasing FSH. And this process continues until you end up with the same total volume of follicle, basically same number of granulosa cells and same estradiol output as you would in a single ovulating animal. So although we weren't able to demonstrate that the selection window or that process in which selection occurs is likely slightly extended in time or is longer in time than what we see in single or control animals. And then the other thing from one of our latest studies that's really intriguing is that when we did the acetylene in replacement with HCG, we saw that just like in single ovulating animals, if you remove LH pulses completely from the picture, you prevent follicle selection. However, replacement of LH activity through HCG in a non-pulsatile manner only allow the selection of a single dominant follicle, which make us believe that either trio carriers require pulsatile LH, or that even the possibility that GnRH-induced FSH pulses are actually required for the selection of multiple dominant follicles. So there's a lot of work still to be done with this model, and also the possibility of extending this model to other circumstances in which multiple ovulations occur, whether it's in cattle, such as the lactating dairy cow model, or even to polyovular species. So with that, I would like to thank some of the members in my lab that have contributed working on this. Folks at UW-Madison, particularly Milo Wiltbank and Brian Kirkpatrick, which started all this work when I was a grad student, and it's been a very interesting ride. And then the incredible number of people that helped through all those years at UW-Madison to do all these experiments. I'm sure I'm forgetting some of them there. And then, of course, our funding sources for that support some of this work, and as well as other things we do in the lab. And with that, I'll thank you for your attention, and I'll be happy to take any questions.